Hello. So the topic is immunology, and we will answer the question from GATE 2021 microbiology section. So the question here asks us to match the immunoglobulin classes with their function. So we have here different kinds of immunoglobulin and their different functions. But to answer this question, we first have to know what are immunoglobulins. So immunoglobulins are basically B cell antigen receptors which are produced by the B cell or the B lymphocytes, which are a class of the white blood cells. B cell antigen receptors are present on the membrane of the B cell. They are attached on the membrane. They have a transmembrane region which helps them to remain attached onto the B cell membrane. Now, this B cell antigen receptor, has a peculiar structure. So, it is composed of two heavy chains. These long ones are the heavy chains which have the transmembrane region, and they also have two light chains which are associated with each light chain is associated with the heavy chain, and they are joined together by the disulfide bridges. So there are three or four they are disulfide bridges which join the heavy chain to the light chain, and one disulfide bridge to join the heavy chain with the heavy chain. So each chain has a variable region as well as a constant region, both the heavy chain and the light chain. The variable region from both the heavy chain and the light chain, they together form the antigen binding site. So there are two antigen binding sites in one immunoglobulin. It will have two antigen binding sites. So another important thing to note about the antigen receptors is that they are very specific. So a B cell will only produce one kind of antigen receptors. So as you can see, this B cell has antigen receptors whose structure are same. So it will recognize only a particular kind of epitope. So what are epitope? Epitope are a small region of the antigen which is recognized by the B cell receptor. So if this whole thing is an antigen, epitope is the small region. So here you can see an antigen consists of various epitopes. One can be circular kind of or conical kind of or others, and this B cell will recognize only one particular kind of epitope. Then it will get activated. After recognizing the epitope, it will get activated, and then it will start secreting these antigen receptors, which are then called the antibodies. These antibodies will recognize those particular epitopes, and that will help in further destroying the foreign particle or the foreign organism. And there are various antibodies like say suppose antibody A, B and C, and antibody A will recognize only one particular kind of epitope. Say suppose this conical one. Antibody B on the other hand will recognize this circular epitope, and antibody C will recognize a different kind of epitope. So each antibody is very specific, and it will recognize only one particular kind of epitope. Another important concept to know here is that this heavy chain and light chain can be digested in various ways. So, this heavy chain and light chain, as you know, they are composed of the disulfide bonds. So, if we perform beta mercaptan ethanol reduction on them, so these disulfide bonds will get reduced, and we will get individual heavy chains and light chains. But if we perform pepsin digestion, so pepsin is an enzyme which cleaves after the disulfide bonds. So we'll get this kind of structure, which is called the FAB prime 2, in which the variable region of the heavy chain and the light chain together, they are joined together like this structure, and we will get two FC fragments, individual FC fragments. And but if we perform a papain digestion, it will cleave above the disulfide bond. So what we will receive is that after digestion is two FAB fragment like this. They are joined together by the disulfide bond, and two FC region which are also joined together by the disulfide bond. So this is what we will get. And here you can see as a three-dimensional structure of the immunoglobulin IgG. Now we will come to the different classes of immunoglobulin. So there are five different immunoglobulin classes, and they are differentiated based on the composition of the heavy chain as well as the number of Ig domains, that is the constant Ig domains. So first of all we have IgG, which is composed of the heavy chain gamma, and it consists of three constant regions. IgM, composed of heavy chain mu, it consists of four constant domains. IgA, composed of heavy chain alpha, consists of three constant domains. IgE, composed of heavy chain epsilon, consists of four constant domains. IgD consists of heavy chain delta, and it has three constant domains. So, and each of these immunoglobulin classes or the heavy chain can associate with the light chain kappa or lamin. So light chain is common, and but it is the heavy chain which is the differentiating factor. And also there are some of the immunoglobulin consists of a J chain or a joining chain. These are present in IgN as well as IgA antibodies only. And why are they present? Because IgG, D and E, which do not consist of a J chain, here you can see IgG, E and E do not consist of a joining chain, they exist as monomeric form only. IgG, D and E. But IgA and IgA, as they consist of a joining chain, they can form higher molecular weight structures, and IgA can exist as a dimer in this form where the two monomers are joined together by a joining chain. IgM can exist as a pentamer where the five immunoglobulins are joined together by the disulfide bond as well as the joining chain over here. So now we come to the major antibody functions. So different classes of antibodies have different biological functions, and their serum levels are also different. So IgM is present at 5% serum level, and it can exist as a monomer as well as a pentamer. It is membrane bound on immature as well as mature B cell. It is the first antibody produced in response to antigen. It is the first antibody produced in the fetus. So these 2 points are very important. It is efficient in binding antigens with many repeating epitopes such as viruses, and uh, it functions in classical complement activation. So there are these 3 points which are important. IgD is present at a serum level of 0.3%. It can exist as a monomer. It is membrane bound immunoglobulin on the surface of base cell, but its biological function is not. Next, we have IgA, which is present at a serum level of 7 to 15%. Its structure, it can exist as a monomer as well as a dimer because it has a joining chain. It is a predominant antibody class present in the secretions. What type of secretions? We can have saliva, we can have tears, breast milk, and mucosa. So whatever mucosal secretions are there, then there this antibody IgA will be present. And it is the first line of defense of infection against the microorganism, because the first site where the microorganism comes in contact with the body is the mucosal sites like the nose or the nasopharyngeal region. Whatever site they are the first inhabited by the microorganism, and since IgA is present there in large quantities, so it provides the first line of defense against the microorganism. IgA is present at a serum level of 85% in a monomeric form, and it is the most abundant class of antibody with four sub isotypes IgG1, 2, 3, and 4, and it can cross the placenta, so it provides immunity to the growing fetus. And it also helps in opsonization. What is opsonization? Opsonization is basically coating the foreign particle with antibodies all over the uh, like the foreign particle is fully covered with the antibodies, and this helps it this helps it the phagocytic cells to recognize the foreign particle, which in turn can phagocytize these opsonized foreign particles. IgE is present at the serum level of 0.02% in a monomeric form, and it is important because it provides defense against parasite infections, and also present it is associated with allergic reaction or hypersensitive reaction, and it is present in the tissues. So based on all this information, we can now answer the question. So So we have here this question like IgE, IgG, mRNA, and the functions are protects the feces. First, antibody produced in response to infection. Ig, then we have provides localized protection to mucosal cells, mucosal surfaces, mediates allergic reaction, and directly lyses the cell. So IgE, as we read, it is associated with allergic reaction. So it is the first option, that is one is S. So here one S is correct only in this option. So in this way you can, like, in 1 second only you can answer this question, but we for explanation purposes I will go through each and every option. IgG, it protects the fetus as we read here, so the second option will be P as it is here. IgM, it is the first antibody produced in response to infection, and number 4 will be it provides protection against the mucosal surfaces. So 4 and 3. So the answer of this question will be A. Option A. Question number 14. Answer is A.